Welcome to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from across the schools and departments of the University of Cambridge, reflecting the pioneering work and diverse interests of members of Cambridge Neuroscience. Cambridge Neuroscience is currently going through quite a detailed consultation process to develop 6 new themes for the research we do here. Each of the next 12 talks will come from one of the 6 new themes, 2 from each. For more info on the themes and the talks covered in this series, please follow the links below and follow us on Twitter at CamNeuro. Today we are delighted to welcome Professor George Malliaras. George is the Prince Philip Professor of Technology at the Department of Engineering here at the University of Cambridge. His research focuses on organic electronics and bioelectronics, and today he's going to talk about electronics and the brain. Welcome, George.
So, hi everyone. Good morning, good afternoon, good evening, depending on where you are. It's a great pleasure to be with you. We'll talk about bioelectronics or using electronics to interface with living systems, with emphasis on the central nervous system. The starting point for the talk is as follows. Why do we want to do this? Why do you want to take electronics and interface them with biology? It's to help folks who suffer from neurological disorders. The observation is that neurological disorders are the major today, the leading cause of disability worldwide, with over 270 million disability-adjusted life years lost to stroke, migraine, dementias, epilepsies, injuries of the brain and the peripheral nervous system. So it's a major societal problem that calls for a solution, a technological solution. Unfortunately, the solution for this is not going to come from big pharma. There is a trend that's indicated here that is called Irum's law that tells that drug discovery is becoming slower and more expensive with time. Irum is more spelled backwards. In electronics, there is the so-called Moore's law that says that every year when you buy a new computer, it's going to be cheaper and faster than the previous one, while in pharma, it's the opposite. It's getting slower and more expensive. So the likelihood that you're gonna get new drugs for neurological disorders is small. However, there's good news, and the good news comes from the field of bioelectronics and what is called today bioelectronics medicine. So look at this video here. This patient has been implanted with an array of electrodes that sits deep in the brain in a structure called the subthalamic nucleus. And through electrical stimulation, you can see a major amelioration with a flick of a switch of the tremor that he has. And this deep brain stimulation, as it's called, has been used quite extensively in motor neuron disorders, also in epilepsy, where it has been successful in reducing the frequency and the intensity of seizures. It is also being tried for neuropsychiatric disorders, such as obsessive compulsive disorder, depression, also being tried for obesity and a couple of other conditions. So fantastic news. Arguably, this goes back to the work of the pacemaker in the late 50s, the development of the first fully implanted pacemaker at Karolinska Hospital, which today is implanted to over 600,000 patients per year. A decade later, we had cochlear implants for helping people who are deaf. Then a few years later, spinal cord stimulation for chronic pain. Then the vagus nerve became a target of investigation for epilepsy and some other conditions. Some other peripheral nerves were also targeted. And then in the early 2000s, interest shifted towards the brain with deep brain stimulation for Parkinson's, obsessive compulsive disorders, and others. Then we saw retinal prosthesis slowly developing, some other stimulators, and more importantly, a trend which is recent is to cuff peripheral nerves and stimulate them as a way to expand the therapeutic spectrum beyond neurological disorders. So people envision autoimmune disorders being treatable with electrical stimulation. So super exciting news. Where's the catch? Why don't we see this deployed to patients at scale yet? There are fundamental and technological reasons for this. Fundamental reasons is that the fundamental reason is that we do not understand how the brain works. There are over 80 billion neurons in the human brain. It's a very complex machinery. And though we understand how neurons process information, fire action potentials that propagate down the axon, and then either excite or inhibit the postsynaptic neuron. These are things we understand in a quantitative fashion. We do not understand how we go from that to behavior. In order to understand more about this, electrophysiologists are using different configurations of electrodes placed on the scalp, on the surface of the brain, or penetrating the brain to monitor neuronal activity and correlate it with behavior as a way to learn the neural language. And vice versa, we do not understand the mechanism of neuromodulation. So what happens when you stimulate with an electrode that's implanted in the brain? Why do we change behavior? And what changes occur on the long term in the neural system? Now, part of the reason why we don't understand this is because our technology is limited. The signals that we are looking at, especially the less invasive you get, the further away from the inside of the brain, whether you're sitting on the cortex or on the scalp, signals get smaller and are hard to detect. The environment is hostile to electronics. When you put a probe in the brain, is bad both for the brain and for the probe. A lot of electronic materials will corrode in cerebrospinal fluid. Obviously, it's a highly invasive thing when you put electrodes in the brain, and often multiple surgeries are required to replace batteries or adjust the position of a probe. So it's a great field to be a technologist in. New technologies are needed in order to address these limitations and enable a better understanding of the brain and bioelectronic medicine as a field. So what I want to do today is to talk about three topics, so illuminate this field of bioelectronics from three different angles. The first one, we will use materials that are called mixed electronic ionic conductors, and I will explain what that is. And we'll show that when you use this as electrodes, then you can improve the coupling between brain and electronics. The second example is a device that does precise drug delivery in the brain, and I will explain why that is important. And finally, I'll talk about electronics that change shape as a means to decrease the invasiveness of neurosurgery. So let me cast the problem of bioelectronics in a broader sense and cast it as a materials science problem. So what we're after is interfacing microelectronics, humankind's most developed technology, largest technological achievement, with living systems. And the first thing you notice when you embark on this endeavor is that there are major differences in properties. Biological systems, especially the brain, are made out of soft materials, the consistency of jelly, while microelectronics in their traditional form are made out of mechanically stiff materials. The communication in the components in the living systems, whether these are cells, tissues, organs, is very complex and often not well understood. While here, the communication between these two components takes place by exchanging electrons or holes, something that we understand quantitatively. Biological systems are dynamic. This little fellow will double in size. His brain chemistry will change. While here, electronics are static. They spend their life in the form factor in which they've been fabricated. So what we need to do if you want to enable bioelectronics is to make electronics with properties closer to those of biology. Now, over the last decade, there has been tremendous amount of work in bridging the mechanical properties mismatch. So we have now electronics that are very thin, very flexible, even stretchable, and they can be incorporated or inside the body, eliciting next to zero mechanical loading of the system. So the term imperceptible is used to describe this electronics. So I would argue that the mechanical properties mismatch has been bridged. The next level down is to bridge the communications mismatch. And what you need to do is to get electronics to speak foreign languages. You want electronics to be able to communicate with more complex signals, whether they range from small metal ions, sodium, potassium, to larger macromolecules, to even components of cells. And you want to establish this communication in a bidirectional fashion. You want to pass information from biology to electronics to do diagnosis, and in the opposite direction to effectuate treatment. So let's look at a couple of examples of how we can do this. And this will be in the context of epilepsy, which is a condition that affects a significant fraction of the world population. And about 30% of the patients show resistance to anti-epileptic drugs for reasons that are not understood. In case of drug resistance, and when epilepsy is severe enough to be disabling, resective surgery becomes the next line of treatment, where electrodes are implanted deep in the brain to localize the so-called epileptogenic zone, the region in the brain that gives rise to seizures, so that it can be resected and then the patient is cured. Now, these are typical data that show a seizure occurring in a particular area of the brain in which this electrode is implanted. And in order to make these calls, you need recordings with a very high signal to noise ratio, good spatial resolution, and it would be very desirable to be able to localize epileptogenic zones without penetrating into the brain. So making better transducers, it's a clear need in this field. Now, let's switch to an engineer's view of this problem. What we are trying to do is you're trying to interface ionic charges in the cerebrospinal fluid with electronic charges in your electrode. Neural activity gives rise to ionic currents. So when neurons fire, they disturb the electrostatics of the cerebrospinal fluid, and they put ions in the cerebrospinal fluid in motion. So imagine this ion here oscillating about. And this oscillation is coupled to an oscillation in an electronic charge inside the material. And then you can measure a current in the outside circuit that will tell you that there is a disturbance in the electrolyte that you can correlate to neuronal firing. Now, as you see in this configuration, here we're using silicon, which is the king of electronic materials. There is often an oxide or a nitride layer that insulates silicon from the electrolyte, insulates and protects it from the cerebrospinal fluid. And that spaces away the two carriers and decreases their interaction. So usually people employ noble metals such as platinum to make electrodes. And in this case, ions and electronic charges can come very close at opposite sides of the interface and couple, but still their coupling is two-dimensional. And it has a hard limit, which is represented by the so-called double layer capacitance. Never mind the actual values here. So an alternative approach that is used to enhance the coupling between these two carriers is to use conducting polymers. And again, we're not going to go into details of structure. Consider this as an electronic sponge, as an electronic material that allows ions to penetrate into its volume. Then you can couple volumetrically ionic and electronic carriers and get much stronger interaction between the two. This allows you to build state-of-the-art devices or make devices that have novel and unseen before properties. For example, you can time reverse the process here, and through electrical stimulation of the electronic material, you can eject ions into the cerebrospinal fluid and effectuate drug delivery. We'll talk about that in a moment. Now, the physics of these materials is still a subject of debate, and there is a group within the bioelectronics community that tries to understand how these materials work and make them better. But the good news is that there are already materials that work exceptionally well. So when ions can penetrate into the volume of the material, their capacitance scales with the volume as opposed to just the area that would be the case for a platinum electrode. So by making your electrode thicker, you make it more efficient. You make it couple electronic and ionic signals better. For example, here we're using a film that is, let's say, 130 nanometers thick. And because ions can penetrate in its volume, it would give 2 orders of magnitude larger coupling between ionics and electronics than a film of the same area made out of platinum. Now, this means that you can take this conducting polymer and make it 2 orders of magnitude smaller, so you gain in spatial resolution, and still be able to record with the same fidelity as a much larger platinum electrode. And this is what we've done here. We have microfabricated this electrode. This is a conducting polymer electrode, which is of the order of magnitude, the size of the order of magnitude of the soma of a neuron. And we couple this with very thin and flexible plastic substrates. As I mentioned earlier, these are widely available today, and you can make electronics that conform very nicely. This is an array of these electrodes that sits on the leaf of an orchid. And you see it conforms nicely following radii of curvature that are important for the brain. Now, when this was placed on the cortex of a rat, we were able to record single neuron activity. These spikes here correspond to the chatter of single neurons that are in the third or so layer of the cortex under the electrode, without disturbing their immediate environment. You're sitting just at the surface of the brain. This was great news at the time because it was thought that in order to record single neurons, you really had to penetrate into the brain and sit right next to them. So this was deemed to be interesting enough for translation to the clinic. And here is this array placed on the brain of an epileptic patient, mapping with very high resolution. So here you get the local field potentials that correspond to average spatiotemporal summations of the chatter of many neurons. But at the same time, you can also get spikes corresponding to individual neurons under the electrode. Now, this was a while ago. Since then, we've been working in 2 directions. The first one is rendering these electrodes multifunctional. One thing we did recently was to make them optically transparent, as shown here. So these are in vitro devices, but it's the same process that would lead to an in vivo cortical array. And this can record spikes with good signal to noise ratio. And at the same time, through this electrode, you could do very high resolution, super resolution microscopy. This is a particular technique which is called structured illumination microscopy that looks at markers at a very high spatial accuracy. So you combine the temporal resolution of electrophysiology with the spatial resolution of some optical techniques, and you can learn much more about your system. The next generation of this is in vivo devices that also turn out to be MRI transparent. I will talk about this in a future event. Another direction we're taking with this is leveraging their properties to give good contacts to make small surface electrodes. So this is an example where we have an array of 4 by 4 millimeter scale electrodes that sit on the surface and can measure myography signals during the motion of a finger with unseen before spatial resolution. So here you can follow polarization and depolarization of the underlying muscle at a resolution that was, again, not believed possible with surface electrodes. So far, what I told you is that by using the appropriate materials that can mix in the same layer electronic and ionic charges, you can detect ionic currents emanating from the brain or from muscle with very high accuracy. Now we're going to flip the coin, and we're going to use an electronic trigger in a device in order to release biomolecules into the brain. And again, this is inspired from epilepsy. As I mentioned earlier, you would use anti-epileptic drugs as the first line of defense. However, there is drug resistance. Also, there can be side effects that these drugs have. So when this doesn't work and epilepsy is severe, then resective surgery, where the epileptogenic zone is localized and then resected, is the next treatment option. However, this is not always possible. Sometimes the epileptogenic zone overlaps an area of high functionality, and then the cost-benefit analysis just doesn't work out. So in this case, we advocate localized delivery of drugs in the epileptogenic zone when and where is needed. Now, this is an interesting proposition. Such localized delivery past the blood-brain barrier has been explored in the case of cancer. And the obvious advantage that you gain is, since you're past the blood-brain barrier, you have a much larger gamut of drugs that you could use. And in the case of brain cancer, you would use this post-resection of a tumor to kill the remaining cancer cells. And there are different approaches. You could use biomaterials or biodevices that will resorb oozing out some chemotherapy. This is obviously not suitable for a chronic application. Or you'd use a catheter that provides convection-enhanced delivery of the drug that you want, where you're mostly delivering solvent with a bit of drug in it. And a key disadvantage is that you have to raise the pressure of the target tissue. You need to push quite hard to get the drug in. This causes edemas and also reflow of the drug on the side of the catheter. So again, new technology needed. And we were very excited when over 10 years ago now, we saw a device developed by our friends at Linköping University and Karolinska Institute that effectuates dry delivery. This means it only delivers the drug without the solvent. So the device works based on a conducting polymer, very similar to the one that I showed you for making electrodes, that bridges two reservoirs. In one, you have the drug you want to transport in an ionic form, here as a cation, and the other is the target. And by applying a potential between the two sides of this film, then you can get the drug that diffuses into the film transported through the process that's called electrophoresis, and then it diffuses into the target. And again, the key issue here is that you're not dragging along solvent. It's only the drug molecule that moves and gets delivered. So this seems ideal for the brain, where you don't want to raise the pressure at this reservoir. The Swedes demonstrated that this type of device can work in supplying acetylcholine in the inner ear. So 10 years later, this device has changed form. This is the current version of it. We use a so-called ion exchange membrane. This is a polymer film that consists of polyanionic charge. So you have anions that are bound to a polymer chain, and they're stuck in place. You have a source reservoir, which is filled with a drug of your choice. And again, the drug here is a cation. It has positive ionic charge. And you also make sure that the membrane is loaded with this drug here. And then the target is the brain. If you apply a potential, as shown here, one electrode placed in the source and the other on the brain, you can move the cations through this membrane and get them to release into the brain, while the presence of polyanions on the membrane blocks an ionic charge from going in the opposite direction. So this device acts as a diode and can transport a cationic drug from a source to the target. So this is the physical manifestation of this device, and this is for rodents. We use a U-shaped microfluidic, as shown here, that has physical holes opened up at the apex, and these are coated with an ion exchange membrane. Then we put in the drug solution in the microfluidic, we let it flow, and here we apply a potential between the inside of the microfluidic and the brain. And through the membrane, we get the drug molecules to come out and into the brain, while the depleted solvent just takes a U-turn and then comes out of the brain through this fluidic channel. And this device can also be sensorized with electrodes or biosensors or what have you. We use GABA, and we can just slightly adjust the pH of the reservoir on the probe in the microfluidic channel so that it can become cationic. And then we can pump it by just applying 0.5 volt. You can increase the concentration locally quite dramatically and achieve a therapeutic effect. GABA is an inherent neurotransmitter of the nervous system that represents a brake system for neurons to stop firing. So this was then demonstrated in an animal model where we use 4-AP injected through a micro needle to create the equivalent of a focal seizure. And then we have 3 scenarios where the device is implanted but not turned on. This is shown here. So here we kindle the seizure with 4-AP, and then here is the seizure as it manifests itself. Then in scenario B, we do the same. We kindle the seizure, then it starts to manifest itself, and then we turn the device on, and the seizure goes away. The spikes that you see here are an electrical artifact that we pick up because we're puffing, applying voltage pulses across the membrane to puff GABA into the system. And then a third experiment is when we prophylactically start supplying GABA. Then we try to kindle a seizure, and the seizure just doesn't manifest itself. And if you go to the paper, you'll see that a bit down the road, you can kindle a seizure with 4-AP if you don't supply GABA. So we're acting on the brain only during when GABA is supplied. So this is very exciting because it shows that in principle, you could imagine using this technology to stop or even prevent seizures. And of course, this is in an animal model, but it's taking its way now towards more bigger models, and hopefully one day to the clinic. And then this gets you thinking, okay, what else could I do with this technology? Can I go back to localized delivery as it was used with tumors? And then you realize that there is no one device that can fit all scenarios. For example, in epilepsy, typically you're targeting a small volume, while brain tumors can be quite large. So just that means you need to optimize the device very differently. We built a numerical model for these devices, and we can look at different parameters such as the amount of drug transported, the pumping efficiency. This means how much drug you pump per reverse current that flows into the device, the on-off ratio, how much leakage you have, and all that, for parameters that we can tune, device parameters that we can tune. And you see that there's no single optimum. This one is optimized at the top right. This is optimized at the left side of this parameter space. So the lesson is you need to make bespoke devices for every condition. And this is the one for brain tumors. It looks very different than the one for epilepsy. And we already answered the most challenging question: can we use this to address large voxels? And yes, it's possible, seen here in an in vitro model. Now, with that, let me spend another 5 or so minutes for the final topic I want to talk about today. And I'll go back to this slide. I mentioned that there is a lot of work on bridging the communications gap. I showed you an example where we use mixed conductors that make it efficient for electronic carriers and metal ions to talk to each other. I also showed you a device where if you apply an electrical current, it can supply GABA with good spatiotemporal resolution in the brain. Now, can we start thinking about addressing the dynamic versus static nature of the of implants? And these are 2 examples of why you would like to do that. This is the placement of a cortical array on the brain, and it requires a craniotomy. Here it's a subdural placement, so it requires opening up a big hole in the dura, and then the array is placed. Another example is a process called laminectomy, shown here, that allows a window over the spinal cord. And this is performed to place a paddle type stimulator. So this is an electrical stimulation array that is placed on the spinal cord in order to help folks who suffer from chronic pain. However, this is a brutal operation. It's done under generalized anesthesia, and there is a significant recovery time from that. An alternative technology is to use this percutaneous electrode array. This can be placed on the spinal cord through a lumbar puncture procedure, which is done under local anesthesia, and the recovery time from this is rather small. So patients today face a choice. This device works much better, but it requires a quite brutal procedure. This device isn't as efficient, and also it can shift in space and lose its target, but it can be placed with an easy procedure. So as a patient, which one would you choose? We try to find ways to navigate this trade-offs, and inspiration here came from this work on soft robotics. So this is a structure made out of plastic, which is actuated with compressed air, to change shape and achieve a particular function. Here the function is walking. So this gets you thinking, can we think broadly of electronics as not having a fixed shape, but having a shape that can be actuated to fit a particular demand? And to cater to this, we made flexible arrays with embedded microfluidic channels that can be rolled and then unrolled axially or laterally as shown here. So here's a manifestation of this. You can make a paddle electrode that would normally require a laminectomy to be placed. But here you can roll it up and have it in the shape of a percutaneous electrode that can be inserted in the body with a spinal tap. And then when you insert it, you can deploy it through pneumatic actuation to open up and become a paddle. This is shown here in an in vitro model. And if you use the right type of electronics, then you see that you don't change the electrical properties upon actuation. So this is shown now in human cadavers. This is the procedure for implanting it in the spinal cord. And you see here the device has two markers on the top and bottom part of the device that when it's rolled up, they overlap with each other. And then when you actuate it, they can separate and show you the space that is covered by the paddle. And then this is a video of the same concept on the brain. Here again in human cadaver, you can you do here a burr hole instead of a craniotomy. You insert the array and then you expand it as shown here. These are markers that trace the edges over 2 centimeters. So a quite significant extension with minimal keyhole type surgery. So we're very excited about this. This is also approaching maturity where we start thinking about translation. So with that, I'd like to stop here. I hope I gave you a flavor of how implantable electronics hold considerable promise for not only understanding the brain, but addressing its pathologies. Mixed conductors can enable better coupling between ionics and electronics. And this allows you to make very high resolution electrodes. We demonstrated those on the cortex for recording single neurons without penetrating the brain. This has already transitioned to the clinic. And I talked about surface arrays that can measure cutaneous electrophysiology with very high spatial resolution. Electrophoretic devices were next. We talked about devices that deliver the drug without the solvent. And as such, they're ideally suited for the brain where you don't want to raise the pressure. We showed that in an animal model, they can stop or prevent seizures through the localized delivery of GABA. And then we talked about electronics that can change shape upon actuation, in this case with microfluidics, as a means to minimize invasiveness of neurosurgery, to have a small hole, sneak it through, and then expand it to cover the organ that you're interested in. So let me thank my co-workers, Damiano Barone, a clinician who co-leads the lab, many talented postdocs and graduate students who were cited along the way, collaborators at Cambridge, outside Cambridge, and also very importantly, funding for this work. I have a colleague who says ideas, everybody has ideas. Ideas without funding are hallucinations. So I'm grateful for these funding agencies that allow us to transition our hallucinations into reality. So thank you again for the invitation, and it's a pleasure to be here. I'm ready to take your questions.
Thank you, George, for that really fascinating talk. Now, don't forget to join us next week when we welcome Dr. Amy Orban. Amy is a research fellow at Emmanuel College and at the MRC Cognition and Brain Sciences Unit. Her research uses large-scale data to examine how digital technologies affect adolescent psychological well-being and mental health. Sign up on the links below and follow us on Twitter at CamNeuro to keep up to date with all things neuroscience in Cambridge. See you next time.